Endocrinology Minutes with cyan. Principles of genetics. Heritability means an estimate of the importance of genetic factors to disease causations. Some of the human diseases and phenotypes are Mendelian, which follow Mendelian patterns of inheritance. Other polygenic phenotypes are a result of combined small and additive effects of variation in many genes simultaneously. Here are the criteria which should raise clinical suspicion for a Mendelian disorder. The first, severe, more severe than usual for the disease. The second, segregating, multiple affect family members or family history of consanguinity. The third, syndromic, features such as dysmorphism, developmental abnormalities, unusual biochemical phenotypes, and etc. And the fourth, too soon, which means early age of onset. As shown in the table, most clinically important traits have heritabilities ranging from 20% to 80%. The heritability estimate from any study must be interpreted in the context of the population in which it is being measured, including the historical period and variability in environmental factors such as socio-economic status and nutrition. These factors likely explain the wide range of heritability estimates for type 2 diabetes, ranging from 40% in Finland to 80% in Japan. It is usually not possible to measure or quantify these factors in any individual, but their combined effects can be quantified on a population level as penetrance, which means the proportion of individuals carrying a genetic variant who exhibit the phenotype. Penetrance rises with the age. Carriers of mutations causing multiple endocrine neoplasia 1 have nearly 100% penetrance for parathyroid adenomas by the age 40, but only 20% penetrance at the age of 20. Genetic variants can take many forms ranging from single base change to translocation of entire chromosomes. DNA is subject to other forms of modifications besides sequence variation, such as the cytosine methylation or packaging into nucleosomes with various biochemically modified histones, each of which can alter gene expression and function. Epigenetic modification is a normal part of development and is the reason why different cells have different properties even though they share the identical DNA sequence. A striking example of the effect of epigenetics is imprinting, the expression of a genetic variant in a parent of origin-specific manner. For paternally imprinted genes, the copy that is inherited from the father is silenced, and only the mother's copy is expressed in offspring. Imprinting can affect the impact of disease-causing mutations. Imprinting can also be tissue-specific. The role of genetics in endocrinology. The genetic basis of each heritable endocrine disease is quantified by its genetic architecture, the number of gene variants or genes, their frequency in the population, and their respective contributions to disease risk or phenotypic variation. It is now feasible to identify nearly every genetic variant in an individual's protein coding genes, called whole exome sequencing, or genetic variants in his or her entire genome, called whole genome sequencing. Genetic information and sequencing in individual patients. Some individuals have no apparent symptoms but with an increased risk of disease. Those with a family history of known genetic disease who have not yet been tested, and those without family history who were tested and found with an apparently pathogenic mutation. It is worth mentioning that, even with a mutation conferring several-fold increased risk, the individual is far more likely to remain free of disease. A prudent course of action can be just watchful waiting. In symptomatic individuals, even with a clinical diagnosis, genetic testing can have great prognostic value. Sometimes, a genetic diagnosis can allow a patient to avoid lifelong treatment and testing. Individuals with diabetes from glucokinase mutations, in a kind of maturity onset diabetes of the young, manifest hyperglycemia but are still able to regulate their blood glucose levels so that they neither require hypoglycemic therapy nor are at higher risk of secondary complications of diabetes. Interpretation of identified genetic variants. Identified variants can be classified into three broad clinical categories: benign variants, pathogenic variants, and variants of uncertain significance. Once genetic variants are identified from sequencing or otherwise, they must be interpreted for their impact on health and disease. This interpretation requires the integration of population data to know whether a variant is seen at higher frequency than expected for a pathogenic variant, computational predictions, which can support a benign classification by showing a variant is silent, such as encodes the same amino acid as the reference base, experimental evidence, which can provide evidence that a missense variant does not alter protein function in disease-relevant bioassays, and familial comparisons. For example, the vast majority of variants for dominant disease can be classified as benign if they are also observed in healthy relatives. This is a clinical genetics laboratory report routine. A few websites may be helpful in this procedure, so I listed it on the right side. Recently, a China metabolic analytics project is launched to provide opportunities for novel genetic discoveries of metabolic characteristics and disorders in East Asian groups. More information is included in their website, and you can directly query the mutated gene. Future perspectives and summary. We investigated multiple reasons for genetic variants, namely genetic background, gene-gene interactions, environment, gene-environment interactions, and random chance, in hope to find genotype-phenotype correlations to draw valid and clinically useful connections between DNA sequence variation in human traits and disease. There is often a trade-off between the number of variants identified and the sensitivity or specificity for detecting and calling any individual variant. We anticipate that a genome sequence will become a standard accompaniment to the medical chart. But there is a question: what part of the sequence should we look at? The exome comprises 1 to 2% of the genome, yet contains nearly 85% of known disease-causing mutations. Given current technologies, exome sequencing supplemented with or following chromosomal microarrays is a reasonable initial genome-wide approach. Remember, the resulting genetic information should always be used in conjunction with complementary data like chemistries or imagings and etc. for clinical decision making. Thanks for watching. If you enjoyed this video, just replay it. If you have any questions or suggestions, feel free to contact me through email. If there is the next episode, see you then!